Fisheries, Oceans, and Coast Guard Canada. So yeah, I'm a fish guy. So we'll talk about some of the applications that we have going in Pacific salmon. So right now, for Chinook and coho salmon, these species can be produced in hatcheries. And when they're produced in hatcheries, the adipose fin can be clipped from them. The adipose fin is here. And when the adipose fin is clipped, then you know that the individual has been produced in a hatchery. Now, when the animals are released from the hatchery, they can also be marked with a code-wire tag, which is a small piece of wire that has individual codes on it. So not all fish released from a hatchery carry these code-wire tags, and most of the fish that are released from a hatchery will not carry these code-wire tags. When the fish return and are caught in commercial fisheries or in recreational fisheries, the fisheries are sampled. The fish that contain code-wire tags are identified, and the tags are then recovered from the returning individuals. So what a code-wire tag is, it's a small piece of wire, and that's inserted into the nasal cartilage of fish. This marking technique has been in existence for about 45 years, and it currently forms a component of the Pacific Salmon Treaty between Canada and the United States. And under the terms of that treaty, there are certain conditions that govern the management of fisheries in both countries with respect to Chinook and coho. Okay, so when the animals are released from a hatchery, a code-wire tag can be inserted into a portion of the fish. Okay, so when they return, code-wire tags are detected through various pieces of equipment, depending on the circumstances where code-wire tags, where the animals are. A handheld code-wire tag detector would typically be carried by somebody doing stream walks, where the fish have spawned in a stream, they've died, and the fish have been noted to have a clip on them, or not, and if the fish contains a code-wire tag, the head is cut off, or the nose is cut off, and subsequently sent to a head lab for recovery. Now, if you're on board a ship or research vessel, you can run the fish through a code-wire tag detector. It'll beep, and you can then cut the head off the fish for subsequent analysis. Or if you're at a fish plant, where you load the catch onto a conveyor belt, and the fish that contain code-wire tags are detected, then subsequently the heads are removed, or the nasal cartilage is removed. Okay, so that's the current system that governs the stock identification aspect of Chinook and coho salmon under the Pacific Salmon Treaty. We think there's potentially a better way to do this, and that is the parentage-based tagging technique. What code-wire tags will give you is the hatchery of release, the year of release, and if there's any sort of special release type associated with the hatchery. So we think we can use genetic methods to identify individuals to specific parents. And if we can do that, then we can provide analogous information as coho tags currently provide. We can provide the hatchery of release, the year of release, and any associated release group information. We think we can use SNPs for that, but we will likely require several hundred SNPs in order to provide the resolution that's required for fisheries management among the Chinook and coho populations. So the underlying principle of parentage-based tagging is that if you sample and genotype all of the broodstock at a hatchery, then that will provide the genetic tags so that their offspring from those parents can be identified back to the parents through statistical parentage analysis. So since you only have to sample the broodstock at a hatchery, that parentage-based tagging is a very highly efficient way of marking genotypes for the offspring. For example, a pair of Chinook salmon will produce about 4,500 offspring, a pair of coho about 2,500 offspring. So if you simply genotype those two parents, you can then identify potentially up to 4,500 fish that could potentially return to a single set of parents. So these fish are released as juveniles, adipose fin clipped, and if we sample then the clipped fish that return in the fisheries, we should be able to provide the hatchery and year of release for all the returning fish that are clipped. That's the theory. So to do that, we have worked on developing Amplicon Panels for both species that would target specific segments of DNA that are known to contain SNPs. And we think that this will provide a very cost-effective method of genotyping the individual fish. And at our lab, we have developed two panels, one each for Chinook and coho, where we target up to 400 amplicons, and we typically genotype 384 individuals per chip, but we're hoping to double that shortly. So we have a ion torrent proton that we use, and we use the P1 chip. So basically the workflow is outlined here from start to finish. Library preparation. We have extracted DNA. We then apply our Amplicon panel. We clean up we clean up the amplicons that are produced. We attach the barcodes. Right now we use 384 as I mentioned, but we hope to increase that to approximately 770 per chip. So after we have the library that have been produced, the amplified product binds to the to the beads, and the complementary sequences are extended, and we can then load our chip, our P1 chip, and then changes and the bases are flowed over the chip, and changes in pH are recorded. And that's basically the technology that we use in the lab. So for stock identification, we would use 3 levels of stock identification. First is the parentage-based tagging, where if we know that the fish is clipped that we're trying to identify, we know it comes from a hatchery baseline. And we would then first try to identify the fish to the brood stocks that's been sampled at a hatchery. If we can't assign a clipped fish to specific parents, but we know it, because it's clipped, it has to come from a hatchery, we would do genetic stock identification. And the third level, we would simply run genetic stock identification on both hatchery fish and wild baseline. So there we can look at the origins of fish, whether they've been produced in a hatchery or not. So for Chinook salmon, we currently have an Amplicon panel that targets 390 amplicons, and we're currently tracking 537 SNPs in those amplicons. So we started this project in 2013, where we collected samples from all of the brood stock that were being used in hatcheries in BC for Chinook salmon, where coho tags are currently applied. That was approximately 10,000 fish brood stock. We've sampled on an annual basis since then, and returns in 2015. We have also sampled jacks that have returned. These are the age 2 males that have returned to the hatcheries. So we plan to hopefully demonstrate that when we genotype these jacks, we will be able to identify them to specific parents at the hatcheries. Okay, so far we are on target reads on proton. We have about 75%. The coverage of the average coverage of the SNPs that we're surveying is about 160 times, and we have a no call rate on average of 5% overall SNPs. So here we have British Columbia, and we have the Canada-US border here, Vancouver Island, then animals around here on the east coast of Vancouver Island. Vancouver's here. So okay, so here, these are the areas where we have obtained samples of Chinook salmon from. I'll mention a few hatcheries. This is our Adnarco hatchery up here on the central coast. Here we have the Shuswap, and here we have Lower Thompson, or the Nicola Chilliwack. And we've sampled a number of hatcheries on the west coast of Vancouver Island and on the east coast here. This is Vancouver Island here. So we looked at population structure. And if you're doing stock ID work, doing genetic stock identification, this is exactly the kind of situation where you want to see, where you have a regional population structure. So that all of the populations that we sampled on the west coast of Vancouver Island are in a cluster, those on the east coast, and here on the Shuswap, the Shuswap and the Nicola are part of the Thompson River, which is a main tributary of the Fraser. So it's all good. So right now we don't have any samples to do the parentage analysis, so we've dropped down to level 2 to do the stock identification. So we have 19 populations from which individuals can be assigned. And this is the accuracy that we have in assignment of individuals, where we have a probability of assignment of at least 50%. This level of accuracy we have not seen before in a genetic method of stock identification of Chinooks. So this the amplicon sequencing allowed large numbers of Chinooks to be evaluated, and the accuracy that we have obtained in assigning fish to individual populations has surprised me very, very pleasantly. So the Adnarco was our Central Coast population. Again, we have 100% accuracy. The Nicola is the up in the Shuswap in the upper portion of the Thompson drainage. Again, very good accuracy. Example, Chilliwack is in the Lower Fraser. We essentially have some SNPs that are essentially unique to that population. So we have a very high rate of identification. So we have potential misassignments. Well, these could be errors that we're making in assigning fish, or they could actually be strays. The strays that have individuals that are born in one river but return to another. Salmon typically return to their stream of origin. That's why we have genetic differentiation among populations. But there will be some individuals that will stray from one population to another. We're also doing work on coho salmon. Again, it's a species that's released at a number of hatcheries in BC. We have them on the west coast of Vancouver Island, the east coast, and through a number of hatcheries in Fraser. We started sampling the hatchery populations in 2014. We plan to sample commercial fisheries and recreational fisheries starting in 2017. We have one hatchery where we have sampled the brood stocks in 2006, 9, and 12, so that gives us the capability then to do the parentage assignments. And we have, in that hatchery, we have examined some fish that have returned with Coteau tags so that we know they are derived from the hatchery at Inch Creek. Our Amplicon panel targets 360 amplicons, 440 SNPs. Our on-target reads are about 75%, coverage 130 times on average for each SNP. Our no-call rate's a little higher at 9%. Now, our work on coho is supported through a Genome Canada large-scale applied research program. It's a project that started a few months ago. It's going to be a 4-year program, and we are obtaining funding for our part in that through Genome BC. So we're very appreciative of that support. Again, we have a very strong regional separation of populations. Notice here on the Inch Creek and in the Lower Fraser, where we had the 3 brood years in 2006, 9, and 12. They all clustered together. Norris Creek and Inch Creek are very close geographically. You could consider Inch Creek almost to be a side channel of Norris Creek. So all of the populations from the Lower Fraser cluster, these are some Thompson River. We have good separation between the east and west coast of Vancouver Island and the southern mainland of BC. So this is exactly the kind of situation we want to see for genetic stock identification. For our parentage analysis, we assigned we were able to assign 146 of 211 coho back to the parents for assignment rate of about 70%. We have also done some previous work using microsatellites, and all the assignments agreed with those that we used with microsatellites. The other 30% we couldn't assign, we think largely came from parents from Nourish Creek, and they were not part of the broodstock sampling program. For the fish that we had codeware tags so that we knew that they were derived from Inch Creek, we were able to assign at least 97% of the sample. So again, we were very encouraged by those results. If we're not able to use the parentage analysis, but we simply use the genetic profile of that particular population, again, we had very high rates of assignment. The levels of assignment are really unparalleled in other methods of stock identification based on genetics for Pacific salmon. And I think these may not be misassignments, but these are probably actual strays, legitimate strays into the river. For example, at Nourish Inch Creek, individuals originating from Chehalis or Stave are highly likely, as these two populations are very close geographically to Inch and Stave. So our lab's vision for how we're going to handle PBT application, well, for us, Chinook and coho are the priority. But we can certainly do similar kinds of work on other species. So what we're doing is we're completing the sampling of all broodstock in hatcheries in BC in which codeware tags are currently applied. And we will be sampling returning fish. That will enable us to look at exploitation rates of particular populations in commercial and recreational fisheries, and this is a requirement under the Pacific Salmon Treaty. So we have to show that our genetic methodology can provide the same information as currently available from the codeware tag program. The Pacific Salmon Treaty renews in 2018, and we hope to be able to demonstrate that there may be an alternative to the codeware tag program. Our view is that parentage-based tagging and genetic stock identification will likely supersede the codeware tag program in fisheries management. The reasons that parentage-based tagging enables 100% tagging rate of hatchery juveniles released. Currently, the marking rate, the tagging rate of releases from hatcheries is about 10 to 15% for production facilities. We can easily add more hatcheries to our exploitation rate indicators. Our current number of hatcheries that we use is limited by the costs of buying tags and applying them to the juveniles that are released. One big advantage of genetic sampling is that it's non-lethal. You don't have to chop the head off a fish in order to recover the tag and identify the origin of the fish. In addition, there are a number of ancillary benefits to using a genetic-based method for stock identification, and I've outlined some of them there. Although we're very optimistic about amplicon sequencing and applying SNPs in fisheries management, we think that microsatellites will still be applied in our facility on an in-season basis for fisheries management decisions for the foreseeable future. we analyze on what we call in-season. This is where we need estimates of stock identification 8 to 24 hours after we receive the samples from our clients. And we process about 20,000 fish on an in-season basis during the year. So we don't see amplicon sequencing replacing that for the foreseeable future. And this is our assessment. We think that these genotyping by sequencing platforms will enable us to genotype at hundreds of SNPs at less than, hopefully, the cost of our current microsatellite or other SNP platforms. We think that this will allow us to meet the requirement of a group of people who evaluated the coast-wide code-wire tag program. If there's going to be a replacement, the cost of the coast-wide replacement must be substantially less than the current system. Otherwise, why do it? We think that the parentage-based tagging can provide the same or more information, and we are planning to continue to apply the work to show that rather than as a theoretical concept, it's actually a practical concept. And I must say that that view is not shared by people who spent all of their lives in the code-wire tag program. So I'll wrap it up there. The funding for this work for Chinook has largely been derived from fisheries, oceans, and the Coast Guard Canada. And for coho, same source, but also, importantly, from the Genome Canada Large-Scale Applied Research Project. And I'm open for questions.
Beacham, I actually have a question for you. I'm going to try to get some mics and get other questions. So I actually have a question for Dr. Beacham, because I think that obviously he's blazing quite a new ground. He's going to face the same challenges typically we face whenever we see a new technology trying to displace the entrenched one. I see some additional benefits beyond where you're getting from CWT. What would the benefits be to a hatchery or to the industry in general? So some things like the breeding fitness or the, I forgot what the other one you were saying. There's some additional benefits to it. What would it be for the hatcheries or the breeding in general?
Well, certainly for the hatcheries, we were asked to undertake some work in order to identify the origins of fish in the Strait of Georgia recreational fishery. The Strait of Georgia is adjacent to Vancouver, because our hatchery management group wanted to increase the production from hatcheries that contribute to that fisheries in order to provide greater economic returns to the people of BC. So there are certainly some economic considerations. In terms of hatchery management, we can calculate heritability estimates. If you can identify fish to specific families, you can identify heritability estimates for traits of economic importance. And these would be the age of maturity. There are individuals that mature at earlier age, these jacks. They appear to have a high heritability component associated with them, and some view that we would want to decrease the abundance of jacks in the populations.